Hello, everyone. My name's Arthur Goldsmith. I'm a consultant in anaesthetics and ICU at RHCH. I'm going to talk to you about two urgent public health research studies that I was principal investigator of and still am. The ISARIC CCP study, Clinical Characterisation Protocol, and the recovery study. So we will all know that the COVID-19 pandemic has been one of the largest global health challenges in modern history. Already there are over 160 million confirmed cases in the world, and in the UK it's more than 4.4 million. The death toll is well over 3.3 million and still rising, and that is an underestimate. So what was it like conducting research in the middle of an infectious disease crisis? Certainly challenges. The biggest challenge, of course, was the fact that this was a completely new disease beginning of 2020. And we weren't quite sure exactly how it was transmitted and how it was treated. Not a lot of evidence to support the treatments that were thought possibly may work. So HHFT had quite a problem making policies and service configurations, and you may recall how they were varying from week to week. We were in the middle of all of that, trying to recruit patients, hot patients, on hot COVID wards, and so hence we were having to get hot ourselves. It was difficult when everyone was pretty stressed and really busy to engage people in research when many had little or no research background. We were consenting in PPE, which raises real problems with communication. And our protocols were constantly changing, and they still are. For the ISARIC CCP study, we are currently on version 10.1, and the research for recovery study, we're on version 15. Each time there's a substantial amendment, there are new drugs, and they all have their own side effects and complications. Need to communicate that to everyone, educate, and populate new paperwork, or a lot of work. But with these challenges came opportunities. The fact that everyone was running on adrenaline meant that everyone was awake and really engaged, keen to be helping find solutions. COVID research was a top priority, trumping everything else. And as a result of many parts of the hospital having come to a clinical standstill and many people needing to shield from patient contact, we had more staff that we were able to use. It was an exciting introduction to clinical research, I think it's fair to say, for a lot of people. And another positive was that we had access to drugs that wouldn't have been available outside of a clinical trial. For example, the monoclonal antibody made by Regeneron. Another thing that we introduced was R&D nurses being able to take on the consent for CTIMP. Of course, when there's a lot of activity from us and research, it does provide a focus on what's happening on the wards. And this had positive effects on staffing, knowledge and skills in these areas. So on to the studies now. First of all, the ISARIC CCP study. What is ISARIC? It's the International Severe Acute Respiratory and Emerging Infectious Consortium. It's a global federation of clinical research networks providing a coordinated response to outbreak-prone infectious diseases. Now, about 9 years ago, they developed a standardized protocol that allowed for the rapid and coordinated clinical investigation for any worldwide outbreak. And so when COVID-19 arrived on our shores, we had something up and ready to go for keeping tabs on this. And so we started populating our database from March. This has been an enormously helpful database for informing public health policies, both in this country and internationally. And it also is an open access integrated analysis platform, so that it can link clinical data to a range of studies. And worldwide, over 300,000 patients are in this database. In the UK, over 200,000, and HHFT have recruited well over 1,200 patients. So, background noise, apologies for that. Onto the recovery study. So, the world's largest randomized clinical trial for COVID-19 treatments. It had been highlighted by NerveTag that there were several possible treatments that needed investigation. And so a group based at Oxford University launched this study in very rapid time. It was expedited by all the research infrastructure and was approved in a remarkable 6 days. Most people know that approvals normally take 6 months, or not quite 6 years, but a long time. This was designed as a simple study so as not to overburden frontline staff. Simple in some respects, but not all. Basically, all the hospitals in the UK have been enrolling patients into this. There are about 40,000 patients now been through the recovery study, and we recruited 163 patients in HHFT. So a look at the study design in more detail. The eligible patients were COVID-19 patients admitted to hospital where the medical teams felt that they were suitable for being considered for the particular interventions. There's quite a clever adaptive factorial design in the randomization. So there were these different domains, different arms to the study that patients at initial randomization could be assigned to. If a patient was not suitable to have a particular drug or intervention, you could still randomize them to any of the other arms of the study. And this diagram just gives a snapshot of how things were at one stage in the study. But like I say, it has evolved constantly. So at different times, different arms would be available. And then we collected some simple outcomes. So what treatments did we deliver at HHFT? Here's a summary of this information. As you can see, some patients had just standard of care, some patients had one intervention, some had many interventions. And there was also the second randomization step for patients that were deteriorating. This was for about a year of the study whilst we were investigating tocilizumab. So what happened? The amazing thing was that within 3 months of the study being up and running, we had a positive result. And that positive result was dexamethasone, a cheap and widely available steroid, was found to really make a difference. It reduced death in ventilated patients by a third, reduced death in hypoxic patients by a fifth. So based on these results, one patient had their life saved for every 8 patients on a ventilator that were put on dexamethasone, or one patient's life was saved of 25 patients who were hypoxic on oxygen given dexamethasone. A very powerful effect. There was no benefit for those patients that weren't sick enough to require oxygen. And so this is why dexamethasone isn't recommended for community COVID patients. It was immediately adopted as a standard of care throughout the NHS and indeed worldwide. We had to wait until beginning of this year to get the next positive result, and that was with tocilizumab, an immunosuppressant monoclonal antibody. And this reduced death by absolute risk reduction of 4%. Once again, that was one life was saved for every 25 patients put on this treatment. The other benefits was that it shortened hospital stay and reduced the need for patients to be put on a ventilator on ICU. There were 5 interventions that were found to be not beneficial, and that included hydroxychloroquine, despite what Donald Trump had to say about that. But it was good to know that there was no point in devoting resources and money to these interventions, exposing patients to side effects and complications that weren't necessary. So the negatives, unfortunately, we can't ignore the fact that we were witnessing the Grim Reaper's work whilst we were on the wards. At one point in the second stage, well, actually for quite a lot of the second wave, there were so many eligible patients that we just didn't have enough research staff to recruit them. That was a bit frustrating. We had logistics problems running the study on 2 sites with the monoclonal antibodies being made aseptically in Basingstoke and getting them to Winchester. That was a problem. And randomizations, inevitably, it took all day to get approval for patients to get into the study, and then randomization would be late, and our team would have to end up staying late into the evening sometimes just to make sure that things happen. On the positive, I was working with these lovely people, and we had great team working and great morale, and still have. We got new recruits to the research team, and we really saw clinicians being reinvigorated in the joys of working in research. We found teleconferencing helped linking staff working from home and working on the 2 different sites, and we were able to get consensus decisions, which was always good. It was really positive to be able to offer patients hope when they were desperately unwell on the wards, and being able to say that we were giving them a new intervention. Ultimately, though, the greatest joy was the positive results and knowing that we had found something that was saving lives. So that's all I have time for. My sincere thanks go to all the people involved in this study, a large group of R&D staff, as well as doctors and nurses and pharmacists and technicians all over the hospital. And I must particularly thank my trial co-PIs. Thank you for your attention. Enjoy the day. Goodbye.